This is chapter 7 and deals with microbial biochemistry. So we're going to look at some of the basic chemistry. Chemistry is not a prereq for this course, so don't panic. This is going to help explain some of the basic chemistry and biochemistry that you will need to understand for some of the other parts of microbiology, like physiology, which involves biochemistry, for it to make sense to you. So it's kind of chemistry in a nutshell, if you will. Organic molecules are molecules that contain both carbon and hydrogen. It has to have both of them. Some of the most abundant elements found in living organisms are carbon, hydrogen, oxygen, and nitrogen, also phosphorus and sulfur. There are over 100 different elements. I believe we're at 119 now, maybe 120. The last several ones they've discovered actually are man-made and only exist for fractions of a second. But the ones listed on here, these are naturally found. They are the most abundant ones that we do find. Just so you know, carbon, hydrogen, oxygen, nitrogen, the top four, those are by far the four most abundant ones. Organic molecules, we're talking about combinations of different elements together. Isomers just mean they have the same chemical formula, but they look different structurally. So you might write it out and say, oh, it's C6H12O6, and you have these two different compounds, and both of them are C6H12O6. But when you draw out the structure, it's different. We often say in human anatomy that structure determines function. This definitely holds true in chemistry as far as if you have a different structure, it's going to have different characteristics. So even though you may have the same number of carbon, hydrogen, and oxygens, if the structure is different, you're going to have some different characteristics. We often use the terms of monomer and polymer. Monomer is going to be small molecules. They're the building blocks. And you put a bunch of monomers together to build the larger molecules or polymers. So when you're talking about a large polymer, a large molecule, you'll often say, what is the monomer? What is it made up of? And it's just asking, what is the building block for it? So here are a couple of examples where we talk about how structurally things may be a little bit different. One thing in chemistry is that sometimes you have compounds and you may see it listed as, say, like D-glucose versus L-glucose. One is just a mirror image of the other. Excuse me. So when we're talking about types of organic molecules, there's four main groups of them. You have carbohydrates, lipids, proteins, and nucleic acids. We're going to look at each of these four groups and what they're composed of. Just briefly, carbohydrates, these are your sugars. They are needed for energy. They are needed as structural components. We're talking about at the cellular level. Lipids are your fats and waxes. They also are used for energy. They're also used as structural components. They're also used for insulation, and some of your hormones are lipid-based. Proteins, these proteins basically are involved with just about any single aspect or activity that goes on inside a cell. Enzymes are a type of protein. Some of them are used for structural, some for transport, some for receptors, some for immune system. So they're involved with just about everything. And then nucleic acids, this contains like your genetic information and also RNA that's involved with making your proteins. So if we look at carbohydrates, carbohydrates are your sugars. Carbohydrates are going to have this formula. Basically, for every carbon, there will be 2 hydrogens and 1 oxygen. So 1 carbon, 2 hydrogens, 1 oxygen for whatever you have. So glucose is the most common and probably well-known carbohydrate. That has a formula of C6H12O6. That would be a carbohydrate. If it ends with OS, OSC, that means it's a carbohydrate. So you have glucose, lactose, maltose, sucrose, dextrose. All of those are sugars. Now we further subdivide them as monosaccharides, disaccharides, or polysaccharides. It has to do with how big are they. The smallest ones are monosaccharides. A saccharide is another name for a sugar. Examples of monosaccharides are glucose, probably the most well-known carbohydrate, and then galactose. Disaccharides, things like lactose and sucrose. And then polysaccharides are things like starch, glycogen, and cellulose. So the monosaccharides typically will have, some of them like glucose will have the 6 carbons here. You have ribose, which has 5 carbons. There's a few other smaller carbohydrates that are monosaccharides, but they're relatively small. Disaccharides, basically you've taken 2 sugars and they are connected together. So like lactose, which is commonly referred to as a milk sugar or dairy sugar, is a galactose and a glucose. So you have 2 monosaccharides or simple sugars, you connect them together. And then your polysaccharides are very complex, very large. Things like starch, glycogen. Starch is made in plants, glycogen is made in animals. Glycogen has a very much of a branching pattern. Both starch and glycogen are basically chains of glucose. When you need it, you just pull one of these little rings off at a time. Cellulose, as you can see, is this large compound, multi-layered, that's connected together. Cellulose is the main component in the cell wall of plants and is the most abundant organic compound on Earth. Lipids, these are your fats, your waxes, your oils, things like that. Therefore, they are hydrophobic. That means they don't like water. They don't want anything to do with water. They will not mix with water. This will include things like fatty acids, which typically have long chains. Fatty acids will be classified as either saturated versus unsaturated. Saturated means there are no double bonds. Those long chains are going to be straight. At room temperature, they're usually a solid. Unsaturated fatty acids, they have at least one double bond, and this causes the long chain to kind of have a kink in it. It's not going to be nice and straight. Typically, it will be a liquid at room temperature. Triglycerides are another type of lipid. A triglyceride has a glycerol. Glycerol is a 3-carbon chain. Anything with glycer, chemistry makes sense the way they name things. So glycer means 3 carbons. So glycerol, glyceraldehyde, anything like that is glyceric acid will have 3 carbons. Each of those 3 carbons has a fatty acid chain attached to it. So triglyceride has a glycerol, and then off of that, there's 3 fatty acid chains. Phospholipid is a glycerol, once again, that 3-carbon compound. Now, instead of 3 fatty acid chains, it only has 2, and in place of the 3rd one, it has a phosphate group. You've already seen phospholipids in terms of when we discuss the cell membrane. And then cholesterol is another example of a lipid. And so here are some pictures showing on the far left here is this 3-carbon chain, that is the glycerol. For triglycerides, you have the glycerol, that 3-carbon chain, plus you attach these 3 fatty acids to it. The phospholipid, you once again have the 3-carbon chain up here, that glycerol with the phosphate group on one end, and then off the other 2 you have what it's often referred to as the tails. Those are the fatty acid chains. Now here's an example showing saturated where there's no double bond, so it lies nice and long and straight, versus an unsaturated where right here you have a double bond and it forms a little kink in it. One thing about phospholipids, when they form these bilayers, these 2 layers, The tail portion where those fatty acids are do not like water. That's the hydrophobic portion of it. And so that's why in the cell membrane, where you've got this long kind of like sheet of it, it's a bilayer with the tails facing towards each other. They're going to naturally form that way. If you were to take a sheet of basically these phospholipids and drop them in, say, a water solution, they're going to form what's known as micelles, which are these little circles with the tails all pointing inward, as you see here, to avoid the water. That's kind of what you're seeing if you drop oil in water and it forms like the little circles, because it doesn't want to mix with it. And then this is showing cholesterol over here on the left. Is this 4-ring structure, multiple carbons on it. That helps to stabilize in eukaryotic cells the cell membrane. It's also used in like animals as kind of a template for making some of your hormones, your lipid-based or steroid-based hormones. Hoping is similar to it. This is found in prokaryotic cell membranes. Same purpose is to help stabilize it. And once again, you can see it is a multi-ring structure. There's an extra ring on it versus what you see with cholesterol. Both of those, once again, are lipids. In terms of at the cellular level, carbohydrates would be used first for energy. If they are not present, then, and I'm talking once again at the cellular level, if carbohydrates are not present for energy, then the cell will use lipids. If lipids are not present, so you have no carbohydrates, no lipids, then the cell can use protein, but you're in trouble at that point. You are at technically a starvation level at that point. And even within the carbohydrates, monosaccharides are going to be easier to use. The simple sugars like glucose, that would take priority and would be used first over, say, polysaccharide like starch. So you have your carbohydrates, you have your lipids, and now you have your proteins. Proteins, as I said, are involved with everything that's going on in the cell. The monomer, the building block, is an amino acid. There are 20 different amino acids. Some of these are found naturally, some you make, some you have to get in your diet. The bond that holds two amino acids together is a covalent bond, so you are sharing a pair of electrons. Specifically, it is called a peptide bond. So a polypeptide is a chain of about 50 amino acids. A protein is a really long chain. You definitely have hundreds of amino acids. When we talk about denaturing a protein, you're destroying the structure. So if you destroy the structure, the three-dimensional structure of it, it's no longer going to function. So once again, structure determines function. So this is just showing an example of two amino acids here, and then you form a peptide bond to connect them together. That's what's happening when you're making a protein. With proteins, we talk about the structure of it, and we classify the structure. There's 4 different subgroups of it. There's primary, secondary, tertiary, and quaternary. Primary structure is referring just to what is the sequence of amino acids. Is it alanine, alanine, serine, or is it glycine, serine, alanine? What is the order of those amino acids? That's all it's referring to. The secondary structure is just regional folding of that chain, not the entire thing, but just in certain little areas, which is going to be determined by the sequence of the amino acids. So secondary structure is dependent upon primary structure. In some areas, it's known as what we call alpha helix, which is twisting kind of like a slinky. Another type of secondary structure is what is known as a beta pleated sheet. This is kind of a zigzag, or if you want to visualize it, it's kind of like when you're a kid or even as an adult and you're hot and you have a sheet of paper and you fold it to make a paper fan, and that kind of zigzagging, that's what a beta pleated sheet looks like. So once again, not the entire chain is folded in this way. It's just a small area, and you can have both of these type of structures within one protein, and you can have several of them. You might have a protein that in the chain has 3 alpha helixes and 4 beta pleated sheets. Tertiary structure refers to the overall 3D structure of a protein. For some proteins, this is it. That's all there is to it. Some proteins also have what's known as a quaternary structure, and this occurs if the protein is made up of several subunits. And so the quaternary structure is, how do those subunits fit together to form now the overall 3D structure for the protein? And so this is showing the primary structure, like I say, it's just the sequence of the amino acids. Secondary structure. Here you have shown up here in the diagram the alpha helix, also shown over on the far right, and then along the bottom is the beta pleated sheet. And then the tertiary structure is, how does all this fit together to form a 3D structure? And if the quaternary structure, if that is present for the particular protein you're talking about, is how do all the subunits fit together? How, in this picture, how does the light green fit with the dark green, with the light tan, with the dark tan? How do those 4 units all come together? That would be the the quaternary structure on there. So each quaternary structure is dependent upon the tertiary structure, which is dependent upon the secondary structure, which all goes back to what is the primary structure. And so here you have, once again, just a different way of visualizing it. Sequence of the amino acids, regional areas of folding or twisting, overall 3D structure, and then if you have subunits, how do they fit together? Nucleic acids. A cell would never use nucleic acids as a source of energy. You wouldn't survive that long. So they're never used that way. Nucleic acids, what we're talking about is deoxyribonucleic acid, which is known as DNA, and ribonucleic acid, which is known as RNA. DNA contains all of the genetic information for the cell, versus RNA, which obtains its information from the DNA, and it's going to be involved with protein synthesis, actually making the proteins. So once again, why is it important if you're saying, I don't care about the chemistry, I thought I was taking microbiology, why am I getting this? Because in microbiology, we don't spend all of our time looking under the microscope. Part of the identification of a microorganism, yes, may initially involve the microscope, but after that, a lot of it will be based on doing biochemical tests and analyzing those results. Why are you trying to identify something? Because most of you are going in the medical field. And so what happens is, you have a patient come in that's sick and you know they have an infection. We have to figure out what is causing the infection. If you get the correct medical diagnosis, then you can provide the correct treatment protocols for that particular disease. So that's why you have to know what organism is it that's causing the illness. Then you can correctly treat it. If you make a correct identification of whatever the causative agent is, then you can also not only treat, but you can come up with hopefully some preventative measures to prevent other people from also getting sick. So it's very important to get the correct diagnosis, which involves analysis of biochemical test results. Here's an example of, over here on the left is using a scanning electron microscope to see a bacteria. This is Pseudomonas aeruginosa. It is a gram-negative. As you can see, it looks like a rod. This is commonly found in the soil, but this also can be a problem. It can cause some infections. It's usually what we call opportunistic. It's kind of like given the choice and you're knocking at the door and somebody opened the door for it. One of the problems we're having with Pseudomonas is that it pretty much likes to grow on almost anything, and so there's some issues with drug resistance with this. Now, one thing that's kind of unique about Pseudomonas is that it will often produce a pigment depending on the nutrients available. This pigment here on this plate you can see looks kind of green. I've seen where it looks more of a bluish. We have one that actually looks more like a bluish color in the lab. It kind of looks almost like the bluish color in the background of the picture on the left. It will often, not always, but it will sometimes glow when you shine a UV light on it. So they have some special little things with them and how they break down chemicals. This is another way of just showing different tests that you can run to try to identify certain bacteria. You can grow it on different media, see whether it grows on it, how it grows on there. You can try to extract various products that maybe it made and analyze those to get, once again, that biochemical analysis to be able to identify the organism. So this is just very briefly looking at the biochemistry that's involved, like I say, your organic molecules, carbohydrates, lipids, proteins, and nucleic acids.